Yes, so the seminar today will be given by Simon Alberti. He comes from Dresden, from the University of Dresden, where I think he's a professor. He did his PhD in Bonn in cellular biochemistry. And then he went to, as a postdoc, to the Whitehead Institute at MIT, where he started to work on neurodegenerative diseases. And he has continued, and since he moved to Dresden, he's trying to combine neurodegenerative diseases with biomolecular condensates. So the floor is yours.
Okay, yeah, thank you for the introduction. And so I'm really glad to be here. And so I'm really a biochemist, I mean, as you already said, by training, but we're also doing a lot of biophysics and cell biology, and we're trying to understand these biomolecular condensates and how they form and how they, what their role is in health and disease. And especially neurodegeneration, I mean, I'm going to tell you a story about that today. So first of all, to get us started, so what I want to tell you about today are these types of objects here. So in cells, we have compartments, and many compartments have membranes around them, and some of them you may know. I mean, mitochondria, for example, Golgi apparatus, they have membranes around them. But there are many compartments that don't have membranes around them, and they really have only been discovered or looked at, I would say, in the last 20 years or so. And I'm just showing you a few examples here. So we have these signaling T-cell receptor clusters on the surface of immune cells. We have centrosomes. These are important for nucleating microtubules, which are important cytoskeletal elements in cells. We have these nucleoli, which are important for putting together factories, so these ribosomal factories in cells. We have also these P-bodies or processing bodies, Barbiani bodies, or stress granules. And these are all structures that are very dynamic. They have no membranes around them, and they contain thousands to millions of molecules. So these are very dynamic structures, and for a long time we didn't really know how these structures form. And something that was realized now more than a decade ago is that if you want to understand how these structures form, you have to actually turn to physics. And you have to think about this in the way of phase transitions. You are all familiar, I assume, with the phase transitions of water. So water can be in 3 different states. It can be in a gas-like state, it can be in a liquid-like state, or a solid-like state. And the difference really here is that the interactions are different, right? So the water molecules are weakly interacting, still weakly interacting in the liquid state, but then more strongly interacting. Also more ordered interactions you have actually in the solid-like state. So this is water, but of course, we're dealing with biology. And the important thing or realization is now that also biomolecules can undergo phase transitions, especially proteins can undergo phase transitions. And so what you see here are proteins that are initially well mixed in a solution, in a buffer maybe, and then they demix and they undergo these phase transitions, and then they can form these filaments, or they can form highly ordered crystals. They can also form gels. They can form glasses, which are kinetically arrested states. And they can also phase separate into 2 liquid phases. You have a dense phase and a dilute phase that coexist. And these are all phase transitions. And so we have known this for a while, but I think the difference really here is now that we're realizing that evolution is actually acting on proteins, on the sequence of proteins, to achieve this kind of behavior, and that there's a lot of biology associated with this, but also, of course, diseases, which is, of course, also very interesting. Now, my lab has been very interested in RNP granules, which you see here, and you see 3 examples. And RNP granules are granules in cells that contain protein and RNA, so they contain 2 different types of macromolecules. And you heard about these before. These are processing bodies. Here we have these stress granules. These are stress-induced assemblies. And then you have these neuronal granules here in neurons. And as I said, I mean, they contain thousands to sometimes millions of different biomolecules, proteins and RNAs. They're very dynamic structures without a membrane around them. And it's been really a long-standing question, how do these structures actually form? And of course, people have also assigned functions to these structures, yeah? So there's one idea that these kinds of structures here, they are involved in processing RNAs, ribonucleic acids. These are involved in storing messenger RNAs in cells that are stressed, and these are involved in transporting messenger RNAs around in neurons. Now, and we got started on this problem about 10 years ago now, and so we actually focused initially on one protein because this protein resided in these granules. And this protein is called FUS, or fused-in sarcoma. It's actually coming from, I mean, the name comes from a cancer that is associated with this protein. Now, what is very interesting about this protein is that it has this RNA binding domain, so it likes to bind ribonucleic acids, but it also has this so-called low complexity domain. So low complexity means that I mean, normally we have 20 amino acids, but here actually only a small subset of them is used. This is why it's called low complexity. And what is also interesting about this domain is that this domain is intrinsically disordered, so there isn't really any structure to this. Proteins normally are folded, but these kinds of proteins, they have barely any structure. They're very floppy. They're very intrinsically disordered, which is really unusual about these proteins.
Why do you call it a prion-like domain? Isn't my idea prion was folded?
Yeah, so this is for historical reasons. So prions, proteins that can form prions also sometimes have the same sequence. And this is something that we actually also resolved, so that many prion-like proteins that can form these aggregates that are associated with prion disease, they can also undergo phase separation. And there's this link between this. We think the phase separation is the physiological state, and the prion is the disease state.
So there are disordered regions in prion?
They're very disordered. They don't have any structure, but of course they can acquire a highly ordered structure. And that's why, I mean, then in the prion structure, of course, is highly ordered, and it's a template, and that's why this is where the name comes from. But I didn't want to confuse you with this historical. But many people call them prion-like domains. So what is also interesting is that this protein localizes in the nucleus in a healthy individual, but then in an ALS patient, so patients with amyotrophic lateral sclerosis, this neurodegenerative disorder. So Stephen Hawking had this disorder, for example, this famous physicist. And there it aggregates in the cytoplasm. And so we thought, okay, maybe we can also learn something here about the link between these granules and this disease, because these diseases go along with these protein aggregates. So the very first thing that we did was very simple. We put a fluorescent protein on the FUS protein to be able to see it in cells. And when we looked at the cells, we saw these very dynamic structures. You see these here. You can also see them merge and fuse. And here's actually, this is the rendered movie. And so there's very dynamic behavior, which of course reminds you of liquid-like behavior. Yeah. So suggesting that we're dealing with liquids here. And initially, I mean, people really thought granules are solid-like, right? This was really an important discovery. Now then, of course, we wanted, is this ability to form these liquid-like structures, is that actually encoded in the sequence of the protein? And for this, of course, you have to make the protein, you purify it, and then you look at it in isolation, whether it can do the same thing. And that's actually exactly what we found. So in a physiological buffer solution, we found that this protein formed these round structures, and these structures showed this dynamic kind of behavior that we had previously also seen in cells, suggesting that the information for forming these liquid condensates, as we call them now, is really contained in the sequence of this protein. And that was really remarkable for us to see. Now, putting now a long story short, I mean, a lot of people worked on this now, and of course now we know much more. We think now that these RMP granules, they form by the process of phase separation or condensation. And for this to occur, you have to have these proteins. This FUS protein is one of them, with these RNA binding domains, and then you have these floppy prion-like regions, as we just discussed. And then you have these ribonucleic acids, and initially these two components are well mixed in a solution. But then there's a kind of a change. I mean, there can be a change, like a post-translational modification, a change in salt, or a change in pH, and then they demix. And then you have this, these dense liquid droplets coexisting with this dilute phase. There's, of course, rapid exchange between them. But the important thing now is that, of course, these condensates come about because there's interactions So there's protein-protein interactions, there's RNA-protein interactions, there's also RNA-RNA interactions. So there's a whole network of interactions that forms, and that is underlying the formation of these liquid-like dynamic structures. Yeah? And now we think this is the principle of how these RNP granules form. So I told you this protein is also associated with disease. So of course, the next question then that we asked is what happens in disease? And one thing that we observed, I mean, what we're doing here is we're building these droplets again in the test tube. This is in isolation. I mean, actually, when you see this here, the test tube, that means we're doing in vitro experiments, not cell experiments. Yeah, just as a guide, you see either a test tube or you see a cell up there. And what we noticed is that sometimes we had these irregularly shaped kinds of droplets. They were not really round. There didn't seem to be any surface tension there. And also importantly, what we're using here, this is a point mutation from an ALS patient. There's just a single change in this protein. And then we observed this. So these fibrils growing out of these irregularly shaped droplets. So there was a further conversion here from this liquid-like state to this non-dynamic solid-like state, and then we actually got these fibers, of course.
The scale is the same, or this star is much bigger?
Say again?
The scale is the same for the solid one with the spiky stuff?
Yeah, I mean, so this is actually, you see, this is 10 micrometers, right?
Even for the picture you put on top?
Yeah, so it's really the same. So they get really huge, right? And I mean, the interesting thing is that so these kinds of structures, of course, also you find in the brains of patients afflicted with ALS. Yeah, and so what this means, of course, is that that these proteins, such as Fas, can undergo these phase transitions, and so the protein can be in this well-mixed state initially, but then it can demix and form these dynamic liquid-like droplets. But then, of course, what can happen is that you get these more solid-like, gel-like states, and then these solid-like fibers. And what we think is really the case here is that in this case, we're dealing with a pathological problem, with a disease problem, and here we have a physiological phenomenon. And of course, what seems to be the case is that cells have to work against this transition here throughout your lifetime, right? I mean, they really have to put an energy and work against it so this doesn't happen really, and that you get then this pathological state. Okay, so this is all, I would say, like, you know, this is now 7 years old. So this is all stuff that we published many years ago. So then, of course, we wanted to actually see, I mean, so these RNP granules that I showed you in cells before, right? They don't consist of a single protein or a single RNA. They actually consist of many, I mean, dozens of proteins, dozens of different types of proteins, and dozens of different types of RNA. And then the question that we asked, can we now really go in and because we're biochemists, can we rebuild these RNP granules in the test tube in their full complexity? And we decided to focus on these granules, which are called stress granules. And I told you already before that they're thought to be involved in translation regulation, so regulating protein synthesis. So the reason why we focused on these structures is they're very easy to induce. When you just stress cells, I mean, this could be with arsenite, which is a toxin, you put that on cells, and then you can see these structures forming very rapidly in about 20 minutes or so, and you could see that they showed this very dynamic fusion behavior. So again, it looked like we're dealing with a phase separation here, phase separation phenomenon. But what did we know actually when we started this project? What was known was that, of course, I mean, so cells actually, when they produce proteins, they do this on polysomes, yeah? And so you have a messenger RNA here, and then you have ribosomes translating the message. And so then, of course, in the stress, and the ribosomes are dissociating from the mRNA, and then they assemble together with the mRNA into these liquid-like structures. And so what you also, I mean, of course, you have then these RNA binding proteins, you have helicases, you also have signaling molecules, and they all come together to then assemble into these liquid-like condensates. And this is, of course, what we know. And I told you already that they were involved in translation inhibition or regulation. There was also some evidence that they're involved in signaling. And this is what we knew. So, you know, RNAs are being translated, and then they assemble together with all these factors into these structures. This is, of course, a very complex phenomenon, right? I mean, how do you rebuild this in the test tube, right? And we started from genetics because there was some evidence that two proteins, which are actually very similar, they're called G3BP1 and 2, they're actually essential, absolutely required for the formation of stress granules. And you can see this here. When you actually delete these two proteins, you knock them out, you see that these structures, these stress granules do not form anymore. And this was actually for us a very important finding because people had done genetic screens and they looked for proteins that could be important for the formation of stress granules, and they actually knocked out hundreds. But these were the only two where the stress granules were really gone. You know, all the other components, they were still there. They may be a little bit smaller. The morphology was a little bit different. So this was already suggesting that these two proteins are fundamentally important here for the formation of these structures. So what are they, these two proteins? Again, we're dealing with an RNA binding protein here. So the protein has this oligo or dimerization motif. And then again, there's an RNA binding domain here. And there's this interesting acidic stretch, which we will come to in a minute. So this is what these proteins are. And of course, now, the fact that genetically they were so important suggested to us that we're probably dealing here with proteins that are involved in the nucleation, in the initiation of these structures. Yeah. And so, of course, if you want to look at the early structures that are forming, you're looking at very small objects, and conventional imaging doesn't really help you here. So we had to use some new form of imaging, which is lattice light sheet microscopy, where you can see very small objects arising in cells. And I'm just going to run the movie. So we stress the cells, and you can see after, in this case, after 2 minutes already, not after 20 minutes, you can see these little puncta arising, and you see how dynamic they are. They fuse, and they get bigger and bigger with time, and really showing you, okay, this is the phase separation phenomenon really here. And then, of course, we wanted to quantify this, and actually in this case, we're looking at the number of these objects over time, and we saw this is a biphasic response. You have this initial rapid response, and they have a plateau phase, yeah? And so we call this here the initiation phase, and then this here the maturation phase. And I will come back to that, because now we're trying to reconstitute these two phases in the test tube from purified proteins and from RNA. So, of course, as biochemists, what we do, we purify things, we purify proteins. So first thing we did is we purified this G3BP1 protein. And then in the absence of RNA, nothing really happened. This was a little bit different to what we saw with the Fas protein, because the Fas protein can actually also form these liquid structures in the absence of RNA. This one wasn't able to do so, which was puzzling at first. But then when we added RNA, we got these very nice liquid droplets, suggesting that they're heterotypic interactions. So these two components have to come together in order to form these dynamic liquid-like structures. Then, of course, we wanted to know, is there any preference for RNA? Because there are different types of RNA. There's double-stranded RNA, there's single-stranded RNA, there's structured RNA, there's unstructured RNA. So we tested a whole bunch of RNAs. And what we found is that this protein actually likes to bind to single-stranded and unfolded and long RNA. And this actually makes a lot of sense, because I told you about forming this network. Yeah? You have to form a network in order to form these condensates. And of course, a long RNA, of course, has many more binding sites than a short RNA, right? So this, of course, makes sense. What also was really good for us to see is that, so single-stranded and unfolded RNA, this is the type of RNA that is actually released from polysomes during stress, because what ribosomes do is they actually remove structure and they remove proteins, because otherwise they cannot read the message. And so this was really, this fit very nicely with our observation that, polysomes disassemble, and that this protein really liked to bind this type of RNA. So then, of course, next we wanted to understand how is this all regulated, right? I mean, now we can, of course, we can reconstitute the initial step, but how is it all regulated? Now, when you look at these types of proteins, and we actually did this here in the simulation with Rohit Papu, what is really difficult with these proteins is that they're mostly unstructured. Yeah, you see there's this part here, there's a little bit of structure, and this part here is a little bit of structure. But the rest of the protein is actually, again, very floppy. It moves around all the time. And so, of course, to look at that, you really have to use simulations, and this is actually what we did. But one thing that we noticed in our simulation, interestingly, is that this acidic domain, so acidic means there's a lot of acidically charged amino acids here, and this arginine-rich domain, so there's, of course, a lot of positive charges here, they were very often close to one another in the simulation, suggesting that there is an interaction here between this acidic domain and the arginine-rich region. And so, I mean, to make a long story short, I'm not going to show you the data, but so we then came up with this model that this protein can be in an open and a closed state. Yeah? And in a closed state, this comes about, I mean, it's actually a dimer, and the closed state comes about because this acidic region and this positively charged arginine-rich region, they interact with one another. So they're electrostatic interactions that actually close the molecule. But then what happens, of course, is that during stress, the local concentration of messenger RNA increases, of course, and the messenger RNA can actually compete with binding here to this positively charged domain. And so now you have an open state. So the molecule is open, and now it can grab the RNA. And then, of course, now you can form the network. And that's exactly what you need to do, right? You need to form this network in order to form these condensates, these liquid-like droplets. So the protein is able to go from a closed to an open conformation, and that's driven by the local RNA concentration. And we wanted to prove this, of course. And we used a technical trick where we can, I mean, this is putting a disulfide bridge in the protein. We can actually fix the protein in the closed state. And then actually we indeed did not get any condensates anymore. Only when the protein was free to move, we got these condensates, showing you really that this open and this closed state is really important for the formation of these droplets. So with this, I think we had explained what is happening in this initiation phase. And remember, I told you about the initiation and then about the maturation phase, right? And I think this is what's happening, right? So RNA is released, the protein grabs onto it, it opens up, and it forms a network. And this is how you get the condensation started. But now what about the maturation phase? Of course, we know from studies with cells that many other RNA binding proteins then are also coming into these condensates. And so we did a very simple experiment here where we thought, okay, let's form these initial condensates first, consisting of G3BP and RNA. And then we asked the question, are all these other RNA binding proteins then recruited into the condensates? And that's exactly what you see here. You see that these are all known stress granular components, and they're all recruited once we have formed these initial condensates. So then we can basically close, I mean, the model. So just to remind you, so you have these mRNAs in translation, protein synthesis, they are released. This protein forms this network with the RNAs, and then we have now other RNA binding proteins and other molecules coming in, and now they form this mature stress granule here as we see it in the cells. So
there's no specificity of the RNA. Any RNA works as long as it's long enough?
Yeah, there isn't really much specificity indeed here. And I think it's really grabbing onto everything that is protein-free and unstructured.
All these RNA binding proteins would go...
Well, they have some specificity indeed. But the initiator doesn't. So they do have some specificity. I agree with that. But many of them can only come later because many of them come from the nucleus.
The one that starts the process has lots of specificity.
The one that starts the process really doesn't have any specificity.
Zero specificity, okay.
No sequence specificity, really. Which is, I mean, but it makes sense. I mean, physiologically, it makes really sense, right? Because it's a protein that basically receives all the RNA that is released from these translating ribosomes, right? And so it shouldn't have any specificity, actually. It would be a bad thing if it had specificity, I think. Okay, so now given that we can build this in the test tube, can we also learn something about the function of these things? And so what is the function? And we did a couple of experiments. I just want to show you one result here, I think, which is interesting, is that when you actually have RNA alone, yeah, at high concentration, RNA alone actually tends to form aggregates. It just forms solid-like aggregates, yeah? You can see this here, and this is also in the model. So you have RNA-RNA interactions because there's base pairing, there's intermolecular base pairing, and you get these, despite all the negative charges actually, so I mean, they have to be neutralized somehow. I mean, this could be through magnesium ions, but you do get these solid-like aggregates of RNA. But then importantly, when our protein GPBP is around, you do get these liquid-like structures, and we actually looked at the dynamics of the RNA, and the RNA is now very, very dynamic. And it can actually be reused later on when the cells go out of the stress, and then they can use these mRNAs again and translate them. So it's, I mean, we think one of the functions of these structures is to keep mRNAs in a happy state, right? So that they don't aggregate and they can be reused later on after the stress to be translated, yeah? But I want to show you a little bit of also unpublished data today because all of this is published, but now, of course, the unpublished work is always the exciting work also to me. And actually we're also, I think you talked about active emulsions today, right? So now we're going into the active area of active condensates. So, and the question that we asked here is, can this aggregated RNA actually be rescued? Let's assume the protein comes too late and the RNA is already in an aggregate. Can we rescue that RNA? Yeah? And I showed you before that one of the proteins that assembles into these stress granules are actually helicases. So helicases are actually enzymes. One of them is this enzyme DDX3X. And so they can actually remodel RNA structures. Yeah, this was known. And so we purified this protein, DDX3X. And first, of course, we wanted to see, is it active? And it shows this nice RNA-dependent increase in ATP turnover, really showing that it has full enzymatic activity, and the enzymatic activity is driven by the presence of RNA. But then, of course, we needed an assay to look at RNA aggregation. And believe me, no one really studies this, right? I mean, there's almost nothing done right now. I mean, there's like 2 or 3 papers in the whole world, and no one is, people are getting more and more interested, but this is really an area where you have to be inventive, yeah? And so we did a very simple assay here to look at RNA aggregation. So we just took total RNA, you can isolate them from cells, and then you just heat it up and then cool it down, and then you get entangled RNA, which is also an RNA aggregate. And you can follow this with a simple sedimentation assay. You spin the sample, this is the supernatant, this is the soluble, and this is the palatable fraction. And you see when you have total RNA before boiling, it's all soluble, it's all in the supernatant, but after boiling, it's all in the pellet, it's all sedimentable, yeah? And that's a very nice assay to look at RNA aggregation, right? Now, then we, of course, wanted to add our helicase here. And you see, this is our helicase that we purified in increasing concentration. This is the insoluble fraction, and this is the soluble fraction. And of course, we're comparing in the presence of ATP and in the absence of ATP to look at the enzymatic activity. Yeah? And what you can see very nicely is that the protein disappears from the insoluble fraction and it reappears in the soluble fraction in an ATP-dependent manner. So this enzyme can indeed disaggregate RNA. And this was really important for us to see, of course, because it showed that this enzyme has this activity. But of course, in cells, there's very often collaboration between proteins. Now, and we thought, okay, so probably the helicase will work together with this GPBP1 protein that I talked about before, this initiator, and probably you have to have collaboration between the RNA binding protein and the helicase. And so then we did a little bit more complex setup here, experimental setup, where we had our aggregated RNA, we had our helicase, and we have our RNA binding protein. Again, we're comparing in the absence of ATP and in the presence of ATP. I'm going to show you in the absence of ATP. This is just a movie. You can just see the structures that are forming here. And you see that in the absence of ATP, over 10 minutes, this structure is very non-dynamic, right? It's solid-like, and nothing really happens in this movie. suggesting that the RNA is still aggregated and that these proteins can't do anything in the absence of ATP. So now I'm going to play what happens in the presence of ATP, which is really remarkable. I mean, so, within 10 minutes, these two proteins collaborate to turn this solid-like assembly into a dynamic kind of liquid droplet. Yeah? And so, of course, this is now how we think this actually happens in cells, right? So the RNA binding protein work together with these helicases to disentangle, disaggregate RNA, and so to allow it to be reused, of course, later on for protein synthesis. And so we can now add this part here. So, of course, one goal, I think, of these stress granules is to prevent this from happening, because this RNA is lost. You can't lose this anymore in translation. But you have helicases that can come to the rescue together with this RNA binding protein. They can keep everything in a dynamic state, and so you can recover the RNA. All right, so that was the part about activity. And the last thing also with this story that I wanted to tell you is, so why are RNP granules actually linked to disease? I mean, genetically, there's dozens of proteins that reside in RNP granules that are linked, to disease. And we asked the question, why is this the case? There must be something about RNP granules in general, why they are linked to disease so often. And we took a very unbiased approach here. We took a protein, this protein is shown on the left here, this is UBC9. It's not really a disease-associated protein. It has a globular fold. You can see this alpha helix, beta sheets. This is how the protein looks. But what we know is that this protein can actually be, there's a variant of this protein. There's a single point mutation that slightly destabilizes the fold of this protein. It's just slightly misfolding prone, yeah? And this, of course, is mimicking these diseases, because these disease proteins also are known to be, they're known to be misfolding prone proteins. And so what we did here is we asked the question, does this protein go into these granules? And you can see when you have stress granules forming and you have wild type UBC9, you see that this does not happen. It's not enriched. But when we have this variant with a single point mutation, this is actually highly enriched in the stress granules. And this was really important for us to see. This is already a couple of years ago. The other thing that we noticed is that there was very aberrant behavior. These granules that were, of course, liquid-like, I showed you lots of data before that they have these liquid-like properties. They were no longer liquid-like. Yeah? So these aberrant stress granules that contain these misfolding prone proteins, they showed no fusion events anymore, suggesting that they had undergone some change from liquid to solid. Another observation that we made is these granules that contain this misfolding prone protein also recruited chaperones. So molecular chaperones are folding helpers, and they bind to misfolded proteins. And there's actually nice, you see this nice linear correlation here between the amount of the misfolding prone protein in the stress granules and the amount of chaperone. So suggesting really we're dealing with a protein folding problem here, and that these misfolded proteins like to accumulate in these stress granules. This is, of course, all in cells, and this is we had done a couple of years ago. Now, of course, given that we have this biochemical reconstitution system, can we now also really identify what's going on here? So, of course, what we do is we then purify these two versions, the wild type and the variant, and we could show that there's this big difference in stability, right? The single point mutations makes this protein much less stable. You can see this here. This is a way of looking at protein stability. So the protein is very unstable as a variant. And then, of course, we reconstituted our stress granules. We had RNA, we had G3BP around. We also had this UBC9 wild-type protein, and we even had the chaperone now in our mix, right? A very complex mixture. And you can see that the granules, or the reconstituted granules, the condensates look very liquid-like, very normal, okay? But now, importantly, in the presence of this variant, we saw these aggregates arising inside the condensates, and they were positive for HSP27, again, indicating that they are recognized as misfolded aberrant species here. Yeah? So something happens inside the condensates that makes this protein unhappy. It just misfolds and aggregates inside the condensates. And then, of course, we wondered, what is this, right? Is it the RNA component or is it the protein component that does that, right? I showed you this before. This is the mix where we have the RNA and the RNA binding protein together with the wild type protein and the variant, and you get these aggregates arising. You've seen this before. But now with a technical trick, we can actually form stress granules in the absence of RNA. We just use a lot of polyethylene glycol. We force condensation here. So we can form stress granules in the absence of RNA, and then we also do not get aggregates anymore. Yeah? This is suggesting that it's the RNA component that drives the aggregation. So to make a long story short, I mean, there's a lot more data here. But what we think is happening is that the RNA in there, because it's mostly protein-free, it's like a scaffold. And these misfolding-prone proteins, they bind to the scaffold, and they're actually brought into proximity on the RNA. And this gets these aggregates nucleated. And then, of course, once you have the nucleation step, and then you form the aggregates. So this is actually, so I think all of these diseases can be explained by this phenomenon, that there is a concentration of RNA inside these granules, and this, of course, is a problem because it leads to the emergence of protein aggregates. So now we can now add this here. I mean, this, of course, has a function in cells, but especially when you get older, right? And when you actually cannot deal with misfolded proteins anymore and your chaperone system isn't as active anymore, this will happen, and proteins will aggregate inside these granules, and then, of course, the disease starts. Yeah. So, summary. So RMP granules, I hope I could convince you that these RMP granules are condensates. That assembly of these structures depends on heterotypic interactions, meaning interactions between different types of molecules, RNA and protein, and you need conformational changes of the molecule. You can have collaboration of RNA binding proteins with these ATP-driven enzymes to prevent RNA aggregation inside. And the conditions inside RMP granules can trigger protein aggregation. And this, of course, is something that cells, of course, want to prevent from happening. And I think the most important thing for me is that as a biochemist now, we are now finally at the stage where we can reconstitute these multi-component cellular compartments from minimal components in the test tube, right? I mean, we could not do this like 10 years ago or so, but now we have the concept, which is, of course, phase separation, and we're now able really to reconstitute these very complex structures in the test tube. Okay, I would have another story, which is another 5 to 10 minutes. Is that okay?
Yeah, no problem.
Yeah? Okay, good. Because I'm also very excited about this. So I mean, we talked about these stress granules now. Forget about them. We're done with them. Okay, good. But the question that we're asking now is, I mean, given our ability now to really rebuild such complex structures in the test tube, can we rebuild other things? Yeah? Can we rebuild other compartments in the test tube? And one compartment I'm really, really interested in is our DNA damage sites. You know, I mean, DNA damage, again, is very often associated with disease. I mean, cancer arises because you have very often problems with DNA damage. And of course, what is interesting about DNA damage is that it actually takes place again in condensates. And I want to convince you of that. So what you see here, these are actually 3 cells. We're going to cut with a laser into the cell to break the DNA. We're generating DNA double-strand breaks. Yeah? And then you will see that this protein, which we have labeled here with a green fluorescent protein, you will see that this protein is recruited to these condensates. Yeah? So, I mean, we cut with a laser here. You saw that? And now you see these puncta emerging. Yeah? And this protein is recruited to these sites where we broke the DNA. Okay? So we can induce DNA double-strand breaks. And of course, we're now very interested in reconstituting these structures in the test tube. Now, what's going on here? Of course, genetics always helps us, right? So we know a lot about this. But I mean, still, the biochemistry is very poor, right? I mean, we know the players, but we cannot really put it all together. And one thing that happens is, so when the DNA breaks, yeah, you have irradiation, high energy irradiation, the DNA breaks. What happens is that this key molecule is PAR polymerase 1. That is recruited as the first player. And then what it does actually, it makes these elaborate polymers, called PAR chains. And it modifies itself, and it modifies all the neighboring proteins. And this has been known. This has been known to an important initiating step in DNA damage repair. And there's all kinds of factors that are recruited downstream of the activation of these, the polymer formation. Again, we heard about this protein already, this FAS protein, fused in sarcoma. So this also plays a role here in DNA damage. What was also interesting for us is that actually PAR polymerase is the most abundant protein in the nucleus after histones. I learned that recently in a paper, which was really weird. I mean, histones are super abundant. They wrap the DNA. And this protein is incredibly abundant. And this makes you wonder, why is it so abundant? Yeah. So what is this protein, PAR polymerase? When you actually look at the protein, it's a very long protein. It's 1,000 amino acids. And interestingly, here we don't have these floppy regions. We have only folded domains. So that was actually a big surprise to us, because you will see later that the protein can form these condensates again. So yeah, what is this protein doing? When you look at DNA damage, and I showed you this laser cut experiment, yeah, and then you can of course look which factors are recruited after the laser cutting, yeah. And you see that PAR polymerase is really the first one that comes on, and you see it increases, but then it also decreases again. And then you have this FAS protein that I told you about, but which comes a little bit with a delay, okay? So, and why is PAR polymerase able to do this? So PAR polymerase actually, what it can do, it takes this compound here, NAD+, and it turns it into these branched polymers. And these polymers can get super long, right? And it just puts it on proteins, and everything around it is added, these polymers are added. You can also do this in the test tube. You can purify PAR polymerase, and then you add NAD+, and you add DNA, because it has to bind to the double-strand break. And then you can see how PAR polymerase modifies itself with these PAR chains here. Yeah? So these chains are added, and you can see that the molecule is getting bigger and bigger and bigger over time. Yeah? Again, we have an enzyme here which is turned on by binding to DNA, and it self-modifies. So again, we're dealing with an active condensate here, yeah, which is really interesting. So then, of course, we wanted to see what about the specificity of this? And actually, I mean, also, of course, what we didn't yet know, but we observed this. Actually, I should tell you this first. So we mixed PAR polymerase with DNA. We actually saw condensates forming, which was a big surprise to us, because this protein didn't have these floppy structures. But you can see very nicely with double-stranded DNA, the protein forms these liquid-like condensates. And it's very specific. When you use single-stranded DNA, or you use RNA, double-stranded or single-stranded RNA, you can see nothing really happens, and this is also quantified here. So there's a very specific condensate that forms in the presence of only double-stranded DNA. But this is, of course, in the absence of enzymatic activity, so you don't need the enzymatic activity for this condensation behavior. But then we wanted, okay, when PAR polymerase has formed these condensates, can it be active inside the condensates? Yeah. And for this, we used NAD plus fluorescein, which is, of course, now you can see the polymers forming because now you have a green label on it. So these are our PAR polymerase condensates. And you can see that when we add NAD fluorescein, now really the PAR chains are added inside the condensate. So really, the PARylation is happening inside the condensate. So we have an active condensate here where the PAR polymerase modifies itself with these chains. And you can also block this. This is a specific inhibitor that's also used in cancer research, in cancer treatments now, in clinical trials. This is also why this is so interesting to us, this PAR polymerase molecule. So you have an active condensate where PARylation is happening. Now I want to move on to this Fas protein, because I mean, I showed you that Fas is also recruited. Yeah? And what is interesting is now we're looking at cells, and you look at this recruitment of this Fas protein over time, and you can see that after a few seconds, it's recruited to these DNA damage sites. But when you block PAR polymerase, this doesn't happen anymore. So the recruitment of Fas is really PAR polymerase dependent. Now, given that we now can reconstitute these DNA damage sites, these PAR polymerase DNA damage sites, can we also see whether Fas is recruited here? And so, of course, we had to do a whole bunch of controls here. I mean, this is our PAR polymerase condensates, and you can actually see that when we have NAD plus, so we're forming the PAR chains, only then Fas is recruited. It's not recruited when we don't have any energy. Yeah, there's no NAD plus here. So our Fas is only recruited when we have NAD plus, and we can form the PAR chains. Then we also, these are two mutants where we changed the arginines and the tyrosines in the protein. This was known from genetics that then this Fas protein isn't recruited to DNA damage sites in cells anymore. And we see exactly the same phenomenon. So we can really, again, we see in our reconstitution system the same behavior as in cells with genetics, yeah? Showing you that probably we're very close to the real situation here with our reconstitution system. So these are active condensates. They recruit this Fas protein. This Fas protein actually, I mean, I told you before, Fas is an RNA binding protein. But what Fas can also do, it binds PAR, because these PAR chains, they look like RNA. They're very similar in terms of structure. And so Fas can actually bind both. It can bind RNA, but it can also bind PAR, and they actually also compete for binding to it. Good. So then we made a very interesting observation. We did a photobleaching experiment to look at the dynamics of these structures. And so you can see this here. I mean, we're photobleaching PAR polymerase, these reconstituted condensates here. This is in the absence of NAD+, so they're not active. Here we have NAD+, so you're forming these PAR chains. And here we have also Fas around. Yeah? And then here, this is actually, in the graph, you see the difference in terms of the dynamics of the molecules. You see that PAR polymerase alone is not very dynamic. There's not much of a recovery. There's a little bit more recovery in the presence of the PAR chains, but then it's much more dynamic in the presence of Fas. And that was remarkable for us to see, because it suggested that the Fas molecule makes the whole structure more dynamic. Yeah? So it's almost like a lubricant that allows you now to go from a solid-like state to a more dynamic liquid-like state. So why would that be necessary, right? So we think the following is going on. This is our model for DNA damage site assembly now. So you have DNA double-strand break, and we have PAR polymerase sitting on the DNA at a very high concentration, because, I mean, this protein is very highly concentrated. We have a break, and what happens is that this, almost instantaneously, this protein forms a cluster on DNA, and we actually think it also doesn't allow the DNA to come apart anymore. I will show you the data in a second. But so it forms these clusters, and then it, of course, it adds these chains onto it, like these PAR chains. And then, of course, this is, of course, a signal now for Fas, which, of course, likes to bind to these chains, is now also recruited. And now you go from a solid-like, more dynamic, liquid-like assembly. And, of course, you need a liquid-like assembly, because eventually you need to repair the damage, right? And you need to allow DNA repair enzymes to get access to this. And this won't be able if you have a solid-like assembly like here. But, of course, it will be possible if you have a more dynamic liquid-like assembly like here. So we think really Fas is sort of like a lubricant which allows now DNA repair enzymes to get in and then, of course, eventually repair the damage. But what could be the function of this cluster here? That's the very last piece of data I want to show you. And that really we find extremely exciting. And so we did a very cool assay here together with Jan Burgos in Dresden. And so what you can do is actually you have a single DNA molecule on the surface of a glass slide. You can see this here. This is the single DNA molecule. actually lambda DNA on the surface of this glass slide. And you can pin it. I mean, you're strapped out, and the two ends are pinned to the glass slide. And there is a restriction enzyme cut site in the middle. Yeah, so actually, when you add a restriction enzyme, it cuts the DNA into two halves. Yeah, and this is actually what we did here. So you have the DNA, and you flow in the restriction enzyme, and the DNA just does this, right? And now you see, actually, what you see is the DNA from the top, right? So it's just hanging around like this. You see the two anchor points here, and this is without PAR polymerase. So remarkable, when you have PAR polymerase there, the DNA doesn't come apart. Yeah, you see, here is PAR polymerase is actually sitting on the DNA, and it's enriched where the cut site is. Actually, you have two cut sites. This is why you have two of these clusters here. Of course, it's also a little bit at the anchor site because there's actually an overhang, so it binds to that. But the remarkable thing is, PAR polymerase, these solid-like clusters, they don't allow the DNA to come apart. Yeah, so that's exactly what you want to do. I mean, you know, in the field, people always assume the DNA breaks and then magically they find back together. But I think very often this doesn't even happen. They just stay together because there's this molecule that does that. Yeah, and it's forming a tether to tether the DNA ends together. Now, this is, of course, without NAD. So when we now, of course, add NAD, can we also recruit our FAS? And that you can see that here. We have NAD present. Now, of course, it paralates itself. FAS is recruited. And we can, of course, now form the DNA damage sites. And, of course, now we can, I mean, we're looking right now also at the enzymes that come downstream. Of course, this is the next step, right? So, but now I think we can also, after having reconstituted stress granites, we can also confidently say, at least the initial steps of DNA damage sites, we can also now rebuild in the test tube, which is really exciting. Of course, there's many, many downstream steps that we have to go through. It's going to be a 10, 15-year endeavor, I think, until we really manage to reconstitute the whole thing. But that's our vision. We really want to rebuild these structures in the test tube. Okay, summary part 2. PAR polymerase forms clusters on DNA ends. These clusters, we think, glue ends together. They tether them to ensure in synapses. PAR polymerase self-paralates inside these clusters, promoting the recruitment of FAS and other molecules, of course, and the assembly of these very dynamic condensates. And then FAS, PAR polymerase condensates, then recruit these downstream. I mean, I didn't show you this, but we've already done some experiments there as well. So this also is possible, that you recruit enzymes downstream. And I mean, very important point. I mean, I told you before for the stress gradients, we can reconstitute now multi-component structures, but I think now we can even reconstitute active. I mean, these are active structures, right? We can really reconstitute active condensates, and by doing so, of course, hopefully understand how complex pathways such as DNA damage work in, of course, health and disease. And with this, I would like to thank the people involved. I mean, those highlighted here in yellow, of course, are the key players, funding sources, of course, here, a whole bunch of collaborators that I, of course, also want to mention. And yeah, if you have any questions, I'm happy to answer them.